we will start with BioVac. Ibrahim Mohammed will do a presentation on BioVac and where they are. Ibrahim.
Good morning, everybody. Hope I'm quite clear on this early Cape Town morning. First of all, welcome to Cape Town, and thanks so much for joining the conference. I know there's two sessions that I don't like presenting in, typically speaking. Normally it's the one just after tea, and then the one just before lunch, which holds everybody up to go into lunch. So welcome back to the session, and first of all, to the organizing committee, it's an absolute privilege to be speaking here today. So, as Charles has already introduced, I'll be doing the BioVac update, and Charles, I will try to keep to time. So just very, very quickly, as a little bit of a background, really it's always difficult to put presentations like this together, especially when you're just starting on a particular project. But I thought for the purpose of this particular meeting, what I would do is basically to present the presentation around the four primary objectives of this program. The first one being the establishment of sustainable mRNA vaccine capacity, which I think we heard quite a bit of this morning from Martin Frieda, Ike James as well. But within the context of Africa and within the context of the low middle income setting, these are quite often challenging things to put in place quickly. And I think Imelda presented a little bit on that earlier, but I'll just elaborate on it a little bit and start the presentation in that way. And then, of course, the introduction of new technologies, looking at the mRNA particular program, the STAR program, very, very exciting program. I think an exciting program for Africa. And I think that's the thing that we should not lose track of. Africa does not have a lot of drug substance technology transfer happening at the moment. There's a lot of fill and finish opportunities, but specifically relating to drug substance, it's obviously very much at a minimal, and there's reasons for that, and most of that is driven through capacity as well. Strengthening regional and biomanufacturing capacity through workforce development. I think African touched quite a bit on that this morning. I'll try not to steer in that direction, but needless to say, with these types of projects, there's always an opportunity for workforce development, and workforce and skills development in Africa is a key component. It's always lovely seeing everybody at conferences like this. Chepu Chiku, I see in the meeting as well from the CSIR. They talk quite a bit about workforce development, so I'll encourage you to talk to him as well. And then finally, just with respect to the regulatory compliance, I think no vaccine presentation can ever end without just noting where the regulatory landscape looks like. Specifically with respect to South Africa, that's what I'll be focusing on, but then also within the bigger scheme, which is really the region as well. So on to the first topic, establishing sustainable mRNA vaccine manufacturing capacity, really talking about the African landscape. So if you look at the top 8 antigens, this is published through the African CDC. I think a lot of you have access to these types of documents, the PAVM studies, etc., etc. Africa is on an exciting trajectory. I think that's the first thing that we have to note, right? 8 or 10 antigens being targeted, moving the needle really from a 1% vaccine manufacturing capacity, hopefully to what they want to get to, which is about 40%. The job today is not to go through these particular antigens that you can see on your slide, but it's really to point out the fact that even though it's an exciting trajectory, only about 3 of those antigens include drug substance manufacturing. And so again, from a workforce development point of view and from a building capacity point of view, these are important things to note. The other thing to note, though, is that the mRNA vaccine technology platform is targeted for one of those antigens. Now, I'm not going to go into the detail. There's no time today to cover it. But I think the thing that I'm trying to point out here is that within the context of Africa, there's still a huge opportunity to develop drug substance capability further. And the mRNA platform, together with the disease burden in Africa, I think will play a vital role in terms of enabling this ecosystem to happen. And then, of course, you could have the technologies, and I think I just want to congratulate Afrigen. I think they've done an absolutely amazing job at building this base technology from absolutely almost nothing. So I think they've done a wonderful job. We've enabled the technology to transfer at BioVac. We'll go through a little bit of the results in a second, but I think I just wanted to pause there and say that they've done an absolutely wonderful job in getting the technology in an amazingly quick time to where it is today. So big kudos to the Afrigen team, led by Karen. But again, you can have the technology, but without having the sustainable platform and the framework to actually push these products into the market, there's still going to be a problem for Africa. And so I think we're very excited as BioVac, and I think Afrigen shares this excitement. And a lot of players on the African continent, even if you're an emerging player, you will share very similar excitement that, look here, at the moment, the time is ripe for African vaccine manufacturers to come into the market and bring products into the market. And why? Because of the framework on the board here, okay? Coordination, I don't want to spend too much time on, okay? PAVM, there's been a lot of delays in that particular study, but even through the African Vaccine Manufacturing Initiative, which I've been a partner of as well, there is coordination in terms of how to move products and how to coordinate within the context of the continent, headed up by the African CDC through the PAVM study. The big one, which was also mentioned this morning in the introductory meeting, I think it was Sophie, spoke to the financial mechanism. So now you've got the coordination starting to happen on the continent, but even more importantly, from a financial mechanism point of view, you've got the incentivization happening through AVMA, which will again enable lower middle income setting countries in Africa to bring vaccine products in and make it price competitive with the global multinationals. Because without this incentivization, again, it's going to be very difficult for us to enter into the market. And the pool procurement is always a discussion. We have a vehicle to be able to handle that. Let's just call that vehicle UNICEF/Gavi today. But into the future, I think the African CDC, they have plans and ambitions to look at enabling this even further. So that gives you a little bit of a background as to why I think the project is important. But let's talk about the project now for a little bit, right? So the project aims, just very briefly, the project aims to industrialize, the Afrigen process, which is at a 1 liter scale, tech transfer to BioVac, and really look at spending a bit of time over the next year optimizing the process and looking deeply into the characterization of this program. Looking at CPPs, the robustness of the process, and really converting it into a fully fledged GMP technology platform that can be utilized at a 1 liter scale, a 5 liter scale, as well as at a 10 liter scale. The overall timeline for the project runs between January 2024 and then, as I pointed out this morning, comes to a conclusion towards the end of September 2026. I think you've all seen this slide probably in various forms and shapes. The most important thing to note here is that BioVac will start the manufacturing process technology from Afrigen, or we've already received that technology in January this year. We've kick-started activities just about a month or a month and a half ago. And really the point that we were driving towards in terms of a milestone is to be able to get to this tech demonstration at a 1 liter scale. And that will literally be just acknowledging the fact that the technology has been absorbed at BioVac and we've been able to reproduce the technology in a like-for-like way from the Afrigen process. And then, as I've mentioned, over the next 6 to 9 months, we'll go into this in-depth process optimization and also into the pre-validation of the assays. That's quite critical. Assays have a way to delay projects quite often. And so assay optimization is going to be key in terms of how this project moves forward. And then this all culminates in the technology transfer packages, really the documentation that summarizes it all, where key components will also be written up in support of CTA. I think for the wider audience here, probably pre-INDs is the word that you guys use, but really for pre-IND submission into the future as well. Not in particular with this program, with this product, but for products to come into the future. So where are we? Okay, you can have the technology, but if you don't have the facilities, you're back to square one and you can't do anything. So in terms of where we are from a facility development point of view, the R&D facilities at BioVac has always been in a state of readiness to be able to absorb the technology and to do the optimization. I think where a lot of time and effort needed to be spent was really into our GMP facility. And in particular, because our GMP facility will always needed to be utilized basically as a hybrid facility. It's really a multi-purpose drug substance facility that needs to be able to handle not only the mRNA production process, but obviously from a sustainability point of view, we developed it so we could move in, as we call it, in a wheel-in, wheel-out concept, campaign basis, one product to the next product. The first product that was in that facility has actually just gone through a SAPRA inspection, and I'll give you a little bit of an update on that a little bit later on. But needless to say, I think from a design point of view, the facility is done exceptionally well, okay? So it utilizes a mix of stainless steel as well as single-use equipment, and pretty much from December 2023, so all of about a year ago, the facility readiness project was completed, and we're in a state of readiness to be able to take projects, put it in there, and take it to the local regulator for authorization. Yeah, and then, yeah, I hate doing this, but I think quite a few of you will have an opportunity to come to BioVac, I think on Thursday, for a site visit. Some of you will not have an opportunity, I'm assuming, but this just gives you a little bit of insight of what the facility looks like. Fully fledged design facility around bacterial capabilities and mRNA capabilities. And as you can see from the slide, just in terms of a pictorial overview, it's really aiming to be a hybrid facility, utilizing single-use plus stainless steel to be able to achieve GMP manufacturing in the facility. The area at the bottom, which hopefully all of you can see, that's really the bottom center column. That's really the wide open space, the DS area that will be utilized for mRNA production as well. And so that was one component of it. I think the second component, as always, is always the analytical, bringing all the analytical equipment in to be able to do all the testing. All the equipment has now been fully installed, and all the equipment also is fully functional, commissioned, as well as qualified in the AMD laboratories to be able to do the testing. I'll give you a little bit of an update on where we are on that in just a few slides. I think it's very important just to say that, look here, while we have a very modest drug substance capability at the moment, BioVac does have plans to move this trajectory a little bit further in alignment with what we're trying to achieve from a continental point of view, right? So for one of our flagship projects, the OCV project, the cholera vaccine project, we're really trying to move the needle here and to get to about 30 million doses of vaccine by 2030. So as part of the design of this particular new facility, which we don't have all the funding for, don't ask me any questions around this. If you have any difficult questions, Morena's sitting just over there, and he'll be able to answer, I think, most of the difficult questions. But we've designed the facility also with a gray box in mind to be able to accommodate any new drug substance opportunities that come along. And key to this, because of this program and other programs that we're working on as well, is the mRNA concept, where, as you can see at the far right of your screen, the gray box can be utilized for mRNA vaccine production up to 50 million doses as part of the expansion project at BioVac. So hopefully that covers the establishment of the vaccine capability, which I think hopefully we've demonstrated. The facilities are ready, all the equipment are commissioned, they're installed, and we're in a good state now to be able to move forward with respect to mRNA. So what about the introduction of the new technology? I've acknowledged Afrigen, that they've done a great job, but really the proof is in the pudding, I guess, when you look at the details, and we'll touch base on that, and how basically this can be utilized to strengthen regional biomanufacturing capacity. So just very quickly, it's obviously a technology transfer. The sending unit is Afrigen. This is quite boring, I know, but just bear with me. The receiving unit is BioVac. I think the important thing to note here is really all the work that goes into a tech transfer, right? And I think what I'd like to highlight here is also the MPP team, who's been working very, very closely with us. So that's really the MPP CMC team, right? In terms of the pre-evaluation of the project. So that includes the pre-tech transfer site assessments. I think this was very important to acknowledge gaps that existed and to be able to close those gaps. The training at Afrigen for 2 weeks that took place, I think, January, February this year. I think it was a very comprehensive training, as well as the stepping stone towards success on this particular project. And then also, before we jump straight into the 1 liter batches, we thought it would be a good idea just to maybe start at the 1 mil scale and then to move to 100 mil scale, just to get our feet on the ground and just to get a feel for where the technology is sitting, because obviously it's a new set of people. They've got a lot of drug substance experience, but they don't have a lot of drug substance mRNA experience. So in terms of overall where we're sitting at the moment, facility readiness has been completed, all equipment has been procured. The tech transfer demonstration batch, the 1 liter batch, is happening this week as we're speaking. All the 100 ml batches has been completed. So please hold thumbs, fingers, whatever is applicable. And then beyond that, we'll go into the optimization, the characterization, and effectively after that, the scale-up activities. Everything, most importantly, remains fully on track. I'm not going to spend too much time on this particular slide. It just gives you the overall time view in terms of 2024 activities all the way through to 2026. I think the milestones are really the key points to take note of over there. The first milestone, as I've said, always being optimization of the process, getting to higher titers, especially on the mRNA type vaccine project where, the vaccine is probably not the cheapest vaccine in the world. mRNA vaccines in general are not the cheapest vaccines. And so really going into an optimization study to be able to improve titers is going to be critical, I think, in terms of the progress of this program. And then analytical method pre-validation, getting the assays to a pre-validated state so that, in terms of tech transfer into the future, there can be an easy transition into the next project. So that's a key milestone. We'll go into the optimization, the robustness testing, etc., as part of milestone 2A. And then I think I've been reminded now quite a few times through technology transfers that we've done in the past, unless you document this stuff and you document it quite well, it's quite meaningless. So I think the milestone package 2, which is really the scale up and the project closeout, including all the documentation, will be a key deliverable that we will have to get to at the end of the project. So very quickly, because I did promise I would stay on time, let me just check where I am. I've got a few minutes, quite a few slides still to go. But just with respect to the method implementation, in terms of the assays, it's looking good. Okay, these are not qualified assays. These are really lab book templates that's just been implemented at BioVac. But as you can see, compendial assays all on track. I mean, I think BioVac has got a very good record with respect to compendial assays, so I think that was quite easy. But more importantly, with respect to the release testing, all of those assays have now been implemented and basically fit for use. Afrigen supplied us with control and reference standards. We've tested it at BioVac, and pretty much there's been very good alignment between the data that they generated versus the data that we've generated. This continues into the DP, where there's good implementation, and the only stuff in yellow is really stuff that's pretty much activities or assays in this particular case that will need to be outsourced to a third party because at the moment BioVac does not have the in-house capability to do it. This is a continuation of the DP, and as you can see again, just with respect to particle size, lipid identity, lipid concentration, everything is implemented. Residual ethanol will be outsourced. We do have the in-house capability, but I think in terms of timelines on the project, we just decided to outsource this particular activity. So where are we with respect to the tech transfer? This really speaks to the 100 ml production scale, and pretty much how I'll end of the presentation. I think Afrigen again has done a great job just summarizing where they were in terms of batches to use as compatibility data for BioVac. Either side of that, there will obviously be a little bit of flexibility in terms of the alert limits on the program. And then just to keep it quite simple, once we go over that particular alert limits in terms of the success, you will get into the failure zone, where potentially it could be a failure if it falls outside of the SME review range. Now, there's quite a lot of detail behind this, but for the purpose of time, I'll not go into a lot of detail. So where are we? Drug substance manufacturing. You see the 1 liter batch, which is basically the African 1 liter batch. This is really drug substance. And then 2 100 ml batches that was also performed at Afrigen. The blue block, which is the blue column, is really the BioVac data that we've generated against our own 100 ml scale batch. And as you can see, with respect to drug substance manufacturing, pretty much everything looks very, very good. We've implemented the process now quite effectively, and without going into the detail, because it will just take too much time, we've pretty much hit the target with respect to each one of the specifications on the screen. The only specification that we did not meet was the final mRNA concentration, and that's purely because we didn't concentrate as low as the Afrigen team did. But that's certainly not a problem. We can just concentrate further at the end of the 1 liter batches, and that should make it within specification. With respect to integrity and size, both specifications were quite easily met at 100 ml scale as well. And then just to close this out, look, with respect to the DP production, we've had 1 crack at it at 100 ml scale as well. All data was in good alignment. Because of the concentration that we took into the particular DP batch, there's a few points that will need to be addressed, and this will be addressed in further studies that we will do at 100 ml scale, hopefully batch 2 and batch 3. And we will hopefully give you an update to that very, very shortly. And you can see a very, very similar trend continuing just with respect to the lipid concentrations, where we are within the SME review in terms of the specification being good, but we'd really like to be totally within the success range, the low and the high limit of the criteria set out by MPP. And so a little bit of work still needs to be done in terms of the actual DP process. From a productivity point of view, that's the actual DS, the quantities, etc., plus, for example, the mRNA quantity that was taken into the encapsulation process. Everything remains fully on track. and the data out of that also looks exceptionally good. So all in all, in terms of where we are right now, I think BioVac is pretty much happy with where the data is standing, and we feel confident moving this process further into the 1 liter batches as well. This is tables that we've just put in place, so this will really be a tracker as we generate the 1 liter data. We'll expand, and as you can see as well from this particular templates, I'm assuming Ike and his team will also be very excited as all the partners start generating data to be able to stack the data up next to each other in terms of getting more and more confidence about the technology as well as the technology transfers. So then just very, very quickly, my final slide, can't leave the regulators out of this particular discussion. So just in terms of regulatory strengthening, I think it's a key component. And really just to say that BioVac walks this path quite tightly. We're very proud to have a very solid and a clear relationship with SAPRA, which is the local regulator. We also get inspected by partner companies quite often, both European as well as American companies. But in terms of the capacity, from a regulatory point of view, we really try and do this through the supply of products into the country, whether that's through local manufacturing or through products that's imported into South Africa. And then furthermore to that, through product development and technology transfers. And this just gives you a little bit of an appetite of what we've done in the past. 9 products really registered in South Africa, 8 technology transfers completed. But I think the most important one is really the one right at the bottom, which really speaks for the first time ever to drug substance manufacturing. And we've just recently been audited through SAPRA for the OCV project, that's the cholera vaccine project. And very happy to report that the audit went exceptionally well. We're still awaiting the final audit report, but just purely from where we're sitting at the moment, it's looking like we'll be going into a phase 3 clinical trial at the start of 2025. So with that, I'll just say thanks to Charles and the team for the opportunity and for you for your attention.
Thanks very much indeed, Ibrahim. That's great. So now we'll move on to biopharma. So Indra Ridiansia, would you like to? Indra, the floor is yours.
Yeah. Good morning, everyone. Thank you, Charles. First of all, I would like to say thank you for the WHO and MPP and the committee for inviting us to give some updates on the implementation of mRNA program at Biopharma. So I would like to start with how biopharmas initiate the work and how we are start building relationship and partnership with the public, private, and academia in order to establish our capability in producing mRNA vaccines. So Biopharma has been quite long experience with the vaccine manufacturing, but mRNA we have zero experience before. So during the COVID-19 pandemic, we have initiative to approach our Indonesian diaspora in Manchester University in order to get some access to the materials, initial materials, and also to the know-how, basic know-how. Then we start working with the mRNA, at least familiarize ourselves with the technology. Then we join with the WHO MPP mRNA Technology Transfer Program as one of the partner, and we would like to establish our capability in producing GMP materials for either phase 1, phase 2, and also for the commercials. So we divided the work into 2 separate groups. The first one is the work package 1 in order to establish our capability in manufacturing mRNA drug substance and the product for at least supporting the clinical trial phase 1 study. And also with the effort from BioVac, we would like to able to be produced at a larger scale in order to make it more for the commercial purposes. But also in parallel, we have explored another also avenue in order to produce the mRNA vaccine. So we're building relationship with a company based in Brussels in order to explore the technology to manufacturing the vaccines. We know that there is 2 different technologies to produce them, but we would like to assess both technology and explore the opportunity with these technologies as well. Since we are focusing more on the drug substance, and after we do some self-assessment, Biopharma is limited by the drug product facility, especially the filling line, because the current capacity is really been used optimized for the polio vaccines. As you may heard that Biopharma is one of the companies that produce the novel oral polio vaccines, and a lot of our drug product is our filling line facility is used for them. So we also have initiative to expand our capacity to produce the drug product, since mRNA can be adapted to different products. So we need more DP facility, and all this initiative is also supported by CEPI. So we have a facility secure the funding from the government and also from CEPI in order to maximize our capability in producing mRNA. Then, based on the support from government, CEPI, and other sources of funding, we try to establish 3 different facilities. The first one is the GLP-1 in Building 40 in order to facilitate us to explore new technologies, new formulations. And this is open for university as well to collaborate in our facilities. And also, at the moment, we are in the process of the qualification of Building 14, which is the GMP facility for phase 1 productions clinical lot materials. Also, Biopharma intends to produce our pDNA locally. So in each facility, we have the facility to produce pDNA either for GMP or GLP as well. As Biopharma quite have good experience in E. coli fermentations, so we're going to proceed with the pDNA productions based on E. coli fermentations. And currently we are in the construction of new brand facility, Building 34. It's a 6-story of facility, and one of the floor is dedicated for mRNA. And this facility can take up until the 50 million doses per year for the drug substance mRNA. And one of the expansion of filling line facility as well will be located in this building, which is in the 5th floor. So regarding the team, because our company is really motivated by making mRNA being able to produce locally, so we are very dedicated to appoint several scientists, administration engineer, and quality people to be dedicated working in this project. This is some update from our side that we have received some introduction training at Afrigen in 2022, and we explained our progress for the first time in the face-to-face meeting in 2023, and we've done also site assessment by the MPP come to Biopharma with CEPI and also WHO. So this is the highlight result that we've done so far. This is the key data that we would like to present as well by our initiative to produce mRNA using technology that we see from Manchester University. We've been able to produce pDNA, at least at the moment, until the 10 liters of E. coli fermentations. We are still working on the yield. At the moment, we are being able to produce at least the efficiency around 40 to 50% based on the mass balance calculations, and we try to improve the yield at the moment. And we also been able to produce mRNA drug substance and drug products at the GLP conditions and try to immunize the mice at whether the vaccines can be able to induce immune response or not. And based on this data, we are quite happy with the result that we've been able to produce at least at the similar level of the commercial one. Then we move on to the next progress, which is the site assessment from Afrigens, from MPP, I'm sorry. So this is the result of our site assessment based on the 2023 results. And at the moment, if we done the self-assessment, we're quite confident that we are progressing toward the favorable state, which is we are ready to receive the technology for phase 1 scale of production. So this is the plan for the tech transfer process. We've given options to produce drug substance at 100 milliliter scales and 1 liter scales. And because we would like to bring up this facility to produce at the large scale, we choose to select the 1 liter scale with our initiative to also perform the 100 milliliter scales. So we will done the small scales, which is up to 100 milliliter at the GLP conditions, and move forward with the transfer at the 1 liter scales. And also, In parallel, we start also scaling up the another method to produce the mRNA, which is the code transcriptions, and to see whether this technology as well could be scaling up at the level that we would like to go. We have identified some of skill gaps in order to receive the technology, and this is the main thing from our human research at the moment. And we have received some funding from internally, our funders, and also from the MPP to how to close the gap. So this is the skill that we have identified, and we will conduct all this training prior the tech transfer process. So talking about the sustainability, we would like also to make our facility, because government already invests a lot and a lot of funded as well. We would like to make how this facility can be sustained for the long terms. So we identified some of the pathogen, one of the consortia that we have built so far in the Southeast Asia. All these vaccine candidates already in our radar, and we have a conversation as well with the developers, and we would happy to produce for them for the GMP scale if the tech transfer already been done. And we also collaborate with the university locally at Indonesia. One of them is to target the HPV and also for the MERS that we are really keen to develop so far. And we also have some conversation with our collaborators, Quantum, in order to be able to produce their candidate vaccine for rabies. So we would like to bring this candidate for the commercials, and also we would like to explore opportunity to develop the product for non-infectious disease targets such as myeloma. Last but not least, we are quite happy to open to any opportunity at the end. So in any kind of form that biopharma can support, we would like to have to support you in any way. So thank you so much. Thanks so much. Thank you.
Thanks so much, Indra. So this is a hybrid meeting. So we're now going to a presentation from Van Son Tran from Polyvac in Vietnam.
Hello, everyone.
Hi.
Could you see my screen?
Can you go to slideshow presentation, because we've got...
Okay. Hi, everyone. I'm Sơn from Polyvac, and we are very pleased to become a partner of the mRNA technology transfer. Today, on behalf of Polyvac, I present to all of you some information about Polyvac as well as about status of the mRNA technology transfer at our center.
Sorry,
My presentation includes 3 sections. The first is introduction about Polyvac, and next is status of the mRNA technology transfer at our center, and the next is the future plans.
Sorry, can you just...
Can I ask you to go to full screen, because you've actually got the... you've got split at the moment.
Okay. Could I reshare? Yeah.
Perfect, Son. Thanks so much.
Okay. Okay. And the first is about Polyvac, and our center established in 1994 with the function of R&D and production of vaccine and biologicals with high quality for prevention of infectious diseases. And we have 3 facilities. The first facility is used for production of oral polio vaccine and rota vaccine, and R&D area is belong to this facility. The second facility is used for production of measles vaccine, measles rubella combined vaccine. And the third facility is island for breeding monkey. Currently, we have 4 nascent vaccine: measles vaccine, measles rubella combined vaccine, rota vaccine, and oral polio vaccine. We also have some in vitro diagnostic kits. Polivac has full ranks of department for vaccine manufacturing facility meeting GMP requirement with total about 145 staff. And up to now, Polivac has many collaboration with international partners such as Japan, the US CDC, PATH, DCVNM, and other partners. Next is the status of the mRNA technology transfer at Polivac. And regard to the mRNA technology transfer program, we have achieved several important milestones as follows. In September 2022, Polivac and MPP signed a technology transfer agreement. In October 2022, we participate in the mRNA technology introduction package in Afrizan. And in December 2023, MPP performed on-site assessment at Polivac, and the results show that Polivac had the capacity to receive mRNA technology transfer at large scale after fixing the facility, purchasing some equipment, and taking part additional training. And in May 2024, we submitted a project to Ministry of Science and Technology for finding fund of materials and consumables. These were approval for contents and waiting for financial approval. In August 2024, we submitted project to Ministry of Health for approval. Up to now, the project is agreed by all Department of Ministry of Health. We assigned 18 staff from different groups for research and production mRNA vaccine in R&D scale. Regarding to facility, we set up now according to MPP experts recommendation. It has 4 main room include room 1 for pDNA production with an area of 18 square meter, room 2 for drug substance manufacturing with an area about 16 square meter, and room 4 for lipid nanoparticle formulation, and the last is QC room for analytical drug substance and drug product. We have some main available equipment for mRNA vaccine manufacturing, such as an autoclave, bioreactor, centrifuge, HPLC system, and some equipment for analytical at Bob and some other equipment. Here in this additional equipment will be sponsored by MPP. In order to support receiving mRNA vaccine technology, Polivac staff have participated some training courses in Korea, South Africa, USA, with content shown on slide. Move on to the section 3, future plans. After discussing with MPP, we make work plan of technology transfer. Right after approval from Ministry of Health and receive the fund, Polivac will perform purchasing and installation equipment and taking part training. The first is hands-on training at Afrizan in October next year, and second is additional training sponsored by MPP. And all training will be done in 2025. For mRNA technology in development of new vaccine, we plan to cooperate with Harvard University Science and Technology to design pyramid, and with National Institute for Control of Vaccine and Biologicals to develop quality control methods. In addition, we are a member of the ANDI Consortium of Hand, Foot, and Mouth Diseases. For sustainable development, we will carry out some activities such as looking for funds for additional equipment of preclinical trials, research and development other vaccine, and also build GMP compliant facilities for mRNA vaccine. And the next activity is contact the government to receive preventive policy and funds, and also make cooperation with domestic and foreign research and organization to promote new vaccine research and development. And based on our current situation and experience of vaccine R&D and production, we estimate that it will be take about 5.5 years to be able to commercialize a new vaccine if we are given mRNA product and have the funds include the following step you can see on slide. Yes, this is all information I want to share to all of you. Thanks for listening.
Thank you so much, Son. That's great. Yes, I noticed you said in there about the importance of having government policies, and we'll come and talk about that later in this meeting. So now we'll move to Inceptor in Bangladesh, and Mohammed Ahassan is going to present to us. Mohammed, over to you.
Good morning, everybody. Charles, can you hear me?
Yes, thank you.
Can you see my slide as well, I guess, yeah?
Yes, thank you.
Hi. Good morning, everybody. My name is Muhammad Mainul Hassan. So at Inceptor, basically, I am heading all the research and development activity for vaccines. So basically, we are the second largest company in Bangladesh in terms of sales volume, and we have more than 10,000 employees, and we have actually around 1,300 products in the market. We have 3 manufacturing sites. So actually, we produce all sorts of pharmaceutical products, including vaccines and biotech products. For vaccine, we started our journey in 2011, and since then, actually, basically, we developed 5 vaccines from scratch. This is meningococcal polysaccharide vaccine, recombinant hepatitis B, oral cholera vaccine, rabies vaccine, and typhoid conjugate vaccine. Among the 5 vaccines, actually 4 vaccines we developed from the scratch. Only oral cholera vaccine, actually, it was the technology transfer initiative from International Vaccine Institute. Apart from this, we have actually many more vaccines in the pipeline. For example, live attenuated dengue vaccine. Actually, basically, this is the very much needed vaccine in Bangladesh. Actually, it's a national requirement. So our team is working hard to bring this candidate to the clinic as soon as possible. So it will go and it will enter the preclinical in January 2000 next year. Apart from this, we are working on HPV, meningococcal conjugate vaccine, pneumococcal conjugate vaccine, live attenuated rota vaccine, and also typhoid tetanus toxoid and diphtheria toxoid. So in terms of mRNA project, basically, we have now 15 scientists working, actually headed by me. They are basically from PhD and MSc level, from the different from diverse discipline like virology, biotechnology, chemistry. We are also actually empowering women and equity. So our scientists first training the introductory training on mRNA technology, and then actually we did another training at Afrigen at Quantum Bison on their mRNA production automated production system. Also, actually this year our scientists also got trained at vaccine manufacturing training at Korea, offered by Wright Foundation. And also, most of our scientists also took the training on biomanufacturing, vaccine biomanufacturing, and good standard practice from actually global training hub for biomanufacturing at South Korea. I think for the future, basically, we have a plan to go for the further training on mRNA technology at Center for Process Innovation UK. That will be hands-on training that will be supported by Imperial College London, and also we'll go for the tech transfer training sometime next year with Afrigen as a part of the technology transfer initiative. So we have almost established actually the facility for R&D facility. All the equipment have been ordered. Most of them has arrived. Some of them is just waiting for receiving. But the GMP facility infrastructure for GMP facility is built with required utilities. Facility will be completely ready by, we hope, by Q1 2026. So this is all the equipment basically. We place the order for the R&D, and most of them has arrived. Now commissioning is on, and few of them still we are waiting for the shipment. That will arrive in our facility soon. So all the equipment has been ordered actually. So basically, since joining this hub, basically we have been focusing on actually for sustainability purpose. We are trying to establish collaboration, how to build, how to develop product quite easy, so quickly to make this program sustainable. So recently we have signed agreement with Quantum Bioscience for co-development of all the reagent for mRNA production. So basically, primarily we are focused on all the enzymes. Apart from, we are also now working. We are working for co-development. Avian influenza. Now that candidate is now they're testing in challenge study now. We are also working on the foot and mouth disease. So basically, we are targeting the animal health and poultry actually. So apart from this, we are also member of the, as you know, the regional R&D consortia. So basically, for the dengue, actually, this consortia is led by IVI and supported by Chulalongkorn University. And this candidate is in, I think the candidate will be ready for preclinical 2026 sometimes. And also we also will be working with the therapeutics HPV. This consortia will be led by Chulalongkorn University. So apart from this, we are also actually in agreement with the Wiseman Lab to support this initiative at University of Pennsylvania. So now we are also trying to work with the Afrigen to develop the RSV vaccine. Actually, RSV vaccine is a much needed vaccine in Bangladesh. Now, apart from this, with Imperial College, actually, we are in agreement to work with their rotavirus vaccine. This is basically self-amplifying RNA candidate. This candidate is basically now pig animal study is going on. So we'll get the data soon. So apart from this, at Imperial, we are also working on to develop a non-LNP based formulation development. So in my next slide, I will talk about a little bit. I have another slide on this. So basically. So basically, to make this progress sustainable, actually, we need to establish more collaboration so that we can bring the product with also for to local and national in terms of mRNA technology. Also, as you know, the clinical trial is very expensive, so we also checking for some support for clinical development for our future candidate. So this is the non-LNP based formulation update. This is basically a polymer based formulation which is called Payball. This is a scalable aqueous only formulation with 100% encapsulation efficiency, and it doesn't require any purification. And it is a cross-link structural stability, which is compatible with lyophilization, also nebulization, and the transfection, intermuscular transfection efficiency is greater than LNP. And the formulation is validated in mice, rabbit, ferrets, sheep, pigs, and macaques. And it is a patented polymer technology currently being GMP scale at Inceptor. We already achieved tenfold scale up. Actually, we are open to collaborate to test various RNA cargo. The good thing is, these lyophilized Payball are stable for 1 month at 37 degrees. So basically, as you understand, we are very much keen to establish a mRNA platform at Incepta. We already, we have set up the RNA lab and working for the GMP lab. So Incepta already itself invested almost, 5 million USD for setup of the R&D lab and GMP production facility. We received some sort of, support from Canadian government, which basically we used for R&D equipment procurement. And for our GMP area, 1100 meter square was built for GMP production facility, whereas all the infrastructure and utilities are in place. Actually, for this initiative, basically, a further external financial support would be appreciated from any funder, I think. So to address financial and regulatory challenge for clinical development, we need to collaborate so that we can sustain and grow better. Finally, I would like to thank all the stakeholder actually who is helping us to establish the RNA platform at Incepta. And finally, thank you very much for listening. Thank you.
Thank you so much, Mohammed. And I just noticed that you mentioned gender equity there, and I know that's very important to some of our funders. So thank you. Right, well, we move on now to Pakistan, to Raim Shah from the National Institute of Health in Islamabad. Raim, the floor is yours.
Thank you much, my name is Shah, and messenger RNA technology over here, using the different vaccine like tetanus, diphtheria, MR, anti-rabies, and allergen sums. So apart from this, we are also dealing with the other vaccines since 1970s. Next, please. Staff working on messenger RNA. The following hierarchy is being followed for the execution of messenger RNA technology transfer. The team lead chief scientist, that is me, and I have three process control scientists, computational scientists, process development engineers. Under process control scientist, they are production scientist, IVT, production scientist, LNP, and quality control analyst scientist. And then production team 1, production team 2, and process testing team, and final product. Next, please. Infrastructures. Although we have allocated integrated space in one of our existing building for dedicated messenger RNA work till the time it will be finalized, the following lab is being used for ongoing R&D activities. Next, please. Infrastructure CGMP. Latest CGMP complies 180-ton HAVOC system is installed to maintain clean area grade B, C, and D, along with air showers, air lock, and ozonization system to prepare aseptic area. The first one is the main lab building, which contains a section for upstream, downstream process, and the other picture is a clean area class C on the right side, and utility rooms that we have producing W5 pure steam and sterile air, and filling areas, filling station class B with laminar flow hoods. Next, please. Equipment. Some of the equipment is already available in the lab, while the equipment specifically required for the implementation of mRNA technologies in procurement phase, which are enlisted here. Nano scaler with the flow meter, HTX plate reader with the Gen 5 software, fragment analyzer, Zeta Sizer with the ZS Explorer software, HPLC CAD, chromatography skit, and TFF. Training. Training received, we have 12 scientists trained from their training on vaccine manufacturing from Ken Iberts, 5 from the introductory training on lab scale messenger COVID and manufactured training from Afrigen Biologics, and we have 4 scientists from GTHB introduction to biomanufacturing. And we have like training in, next please. Yeah. We have training in a plant, training on 100 ml IVT and drug products manufacturing from Frigen, and training on downstreaming process of messenger RNA from Kenai but Island, and training on TFF from Sartorius. Next, please. R&D work. Afrigen COVID-19 plasmid has been received on filter paper, and transformation has been done in E. coli, DH5 alpha, and top 10 cells. Now we are moving on to the linearization of the plasmid for IVT. Parallelly, NIH has also designed the rabies customized plasmid for messenger RNA, which has been validated computationally and in the synthesis phases. Collaboration with the Witwatersrand University to transfer the plasmid template. Raw material and consumable for the lab scale IVT has been produced in all as set to undertake the manufacturing of mRNA in the lab. Collaboration. NIH has initiated collaboration with the local university to submit their research interest in on mRNA. Collaboration with the Aga Khan Hospital to conduct the phase 1 and phase 3 clinical trial. NIH is also approved site for a regulatory body for phase 3 clinical trial and has successfully conducted CanSino Bio COVID vaccine phase 3 trial during the pandemic. Next, please. Plan for sustainability. Funding engage the funds from government source, international grant, and public-private partnership to maintain the operation and capability. Government of Pakistan has also been taken onto the board for the provision of the fund as soon as the DP is developed, also checking for international grant and collaboration. Collaboration engage university to support their research activities on mRNAs, continuous on R&D on existing and emerging disease in the country. Capability building master trainer will train local scientists, researcher, and mRNA process to build skilled workforce that can continue to innovative and support the initiative. Next, please. Question: how long would we need to have our own project ready to sell if we have sufficient fund? The timeline can be quite extensive even with the sufficient funding. Overall, 4 to 5 years are required to have our own product ready for commercialization. Here is a detailed break through down of the process. Clinical trial phase 1, 2, and 3, 24 to 36 months. Regulatory approval will take 6 months. Scale up of manufacturing will take 1 year. Thank you very much. Yes, last.
Thank you very much indeed, Rahim. So now we'll turn to Darnitsa in Ukraine. So Sergei Onishenko, please, the floor is yours.
Thank you, Charles, indeed. I hope you can hear me and I hope you can see my screen.
We can, both. Thank you.
Thank you. Okay, I'll try to be really short. So, quick reminder what Darnitsa is. We are a Ukrainian drug manufacturer. We are extremely new to biologics, but we are extremely old to generic drugs, which we have been doing for almost 100 years. We are a big player in the Ukrainian market. If you go to a drugstore, every sixth pack that you have there will be manufactured at our site. We are launching new products every year. We have 20 plus years of GMP certification from all sorts of authorities, but unfortunately, we are very new to biologics. So what are we doing so far? Well, what have we done so far? We're really focused on the strategy of how do we do the mRNA deployment and biologics and generics at our facility. This is basically a greenfield project, which requires a lot of thought to be put into before huge investments flow into this endeavor. And we are very grateful to WHO and MPP who are supporting us in this activity, who have identified the contractors and the project to do the pre-feasibility study for our activity. We are in this process right away. We are interviewing a lot of stakeholders, both inside the country and outside, trying to come up with the feasible framework for the project to go live. In terms of the tech transfer within the program, we have completed the FTO analysis, which gave us good results, and we did the site assessment of Darnis. Unfortunately, the folks from Geneva couldn't join us in Ukraine, but we did it remotely, and the results were satisfying to us, which proved that we are a capable manufacturer and capable drug maker, but completely incapable of biologics, and this is what we're working on right now. We are very much focused on the team, as I mentioned earlier. This is a huge enabler and to build the capacity within the organization. So we have put together a team of professionals from all different studies, all different spheres on Darnica. And the team is undergoing quite an extensive study plan to build the capacity of biologics mRNA at Darnica. This is just a number of trainings that we have visited and participated in 2024. And as we speak, the head of our project is in Korea right now doing the IVI hands-on training. What's going on right now? The strategy and financing, as I mentioned, we are in the final stages of the pre-feasibility project, which we hope to finish by the end of the year. We are talking to all sorts of international development agencies and funds to, as I said, to structure the framework. We are quite active with the government, trying to pull all possible stakeholders into this activity. Unfortunately, the government is busy with other things. We're in a war. We're trying to defend ourselves again, from Russia. So there are other priorities on the table. However, we think that the further development of the biologics production facilities and the mRNA production in Ukraine is a priority, and we are sticking to it. Once again, the learning and the capacity building block, we have a number of trainings lined up in front of us with the manufacturers within the Vision project, which is another activity which we are engaging. This is all about the capacity building. And the typical question, what happens if we have a commercial viable product? How soon can we bring it online? In a standard scenario, up to 4 years for full facility setup and regulatory approval, then we can sell it to the market. That's it for now. I hope it was short and informative. Thank you.
Thank you, Sergey. It was both short and informative. So yes, thank you. So now we'll move on to the Institute Torlak in Serbia. So Rajna Minic, the floor is yours.
Hello, can you hear me?
Yes, perfect. Thank you. And see your slides.
Okay. Okay, so here I will present. My name is Rajna Minic. I'm from Institute Torlak from the Scientific Research Division. So Institute Torlak is a national manufacturer of high-quality, safe, and effective vaccines, sera, and other immunobiologicals, and it was established by the government of the Republic of Serbia 100 years ago. So other than production, we are also involved in monitoring of infectious diseases, as well as in scientific research and educational activities. Just a second. So Institute is run by acting director, Professor Darija Kisic, and management board and supervisory board, and it is divided into 9 different divisions. among which we have a quality assurance and quality control divisions, and scientific research division, where I am from. Also, we have about 70% women in our workforce of 290 employees. Within the mRNA technology transfer program, we have signed the initial agreement in 2022. Then we have been through site assessment by the MPP in September 2023. We have signed an annex in April 2024, which enabled us to proceed with procurement of equipment, reagents, and consumables, which is currently in progress. And we have been for hands-on training in Afrigen quite recently. So this is our core team, and I must say that from the moment that we have started this project, we have improved in a way that now part of our team is Professor Milan Kojic, who has 30 years of experience in molecular genetics. And we also have some additions to the team which have improved our capabilities. This is our manufacturing facility. Actually, this is the facility where we intend to do first operations on 100 milliliter scale. And we have also purchased some equipment for this process. Regarding the trainings, we have been through several trainings, of which I personally think the training at Afrigen, which was in October, November 2024, so basically last month, was most useful because this is the actual process which we have to go through. Regarding our collaboration with other organizations, so we are collaborating with the Faculty of Medicine, University of Belgrade, where we have a joint project, which has, together with some other projects that we have, enabled us to do some small-scale laboratory operations within the scope of mRNA vaccines. And we are also involved in the Vision project led by the Institute Pasteur Tunis for capacity building in terms of staff exchange. Our operations in the R&D department are the following. So for the project which we have with the Faculty of Medicine, under the idea of Professor Milan Kojic, we have designed the plasmid, so our pDNA for vaccine, mRNA vaccine production, in which we can exchange antigen. So we had produced two plasmids, both on PUC18 and PUC19 backbones, but we have noticed that we have instability in the poly-A tail, which is why we have now transferred to the PACYC 184 backbone. So this work on this new plasmid has also been completed. And regarding our, let's say, interests in the future, so we have looked at the Beacon Intelligence database, basically. So we see that currently there is 11 COVID-based vaccines, mRNA vaccines approved, and one against RSV. And about 1/4 of the clinical vaccines in active clinical trials are self-amplifying RNA. So the majority are non-amplifying RNAs. So we will go for the non-amplifying RNA. And our primary interest is, besides finishing this technology transfer on a 100 milliliter scale for the COVID-19 vaccine, we are also interested in making an influenza vaccine, because the Institute produces already influenza vaccine on chicken eggs. And we would like to have our own vaccine, mRNA vaccine for influenza. So we would do comparative study and know how to proceed further. So We are also interested in producing a rabies vaccine. So we have started some operations on this vaccine with the antigen in the novel pDNA, which we have produced. And we're also interested in the West Nile virus vaccine, as well as in tuberculosis vaccine, because the Institute produces currently BCG vaccine as well. So these are our plans. I tried to be brief, and the majority is in the research. Everything is actually in the research area, and we also are in the process of acquiring all the necessary things which we need to start with the 100 milliliter scale production of the COVID-19 mRNA vaccine. So we are aware that mRNA vaccines are the present and the future for many infectious diseases. And we are very happy to be part of this program. And we are very happy and we are thankful for the funds and the know-how that we have acquired through this program. Thank you very much.
Thank you so much. And we'll end this particular pre-lunch part of the program with another in-person talk from Hiba Wali from the biogeneric pharma in Egypt. Hiba, the floor is yours.
So good morning, everybody, and good afternoon for whom listen us from the other side of the world. And thank you very much for MPP, for WHO, and Afrigen for invitation for such important meeting, which is gonna to highlight what the progress, what is the challenge, and the way forward for the next period, what we are gonna to do in this project. So thank you very much for that. And I'm gonna to start my presentation about introductory about BGB, whom we are, what we are doing with the product, what is the capacity, and brief about BGB product, and then the facility capabilities and team training, and by the end we are gonna to highlight the progress in our project for messenger RNA. For an overview about BGB, it is a brand new facility. It is a state-of-art facility for biopharmaceutical production, specialized in therapeutic recombinant protein as well vaccine. And the main activity, we start designing the facility from R&D, production, quality control, sales and marketing, as well quality control for all activities like monoclonal antibody and vaccine. So when we start thinking about the design of this facility, we put a vision and mission to be a leader in the vision will be a leader in biopharmaceutical company in developing production of therapeutic biological products and vaccine. In order to comply with this vision, we have put a mission to provide a vaccine and biological product of highest quality. How to do that? So we start to think about the design to meet the requirement of international organizations like WHO, EMEA, and FDA regulation. So this is a brand new facility, and it had been designed and started the construction in 2019. And this project or this facility had been worked in two phases, phase 1 for drug product and phase 2 for drug substance. So for drug product, we finalized and we finished the facility for drug product, and we had been visited by many institutes, and this is the outcome of the visit of Africa CDC. So the Africa CDC, they consider BGB is one of the 9 commercial scale vaccine facilities installed, which aim to provide 35% of the continent vaccine by 2030. So this is under the initiative coming from Africa CDC and African Union that by 2040, 60% of the vaccine which is going to be needed for the country, for the African continent, to be locally produced in the continent. So, and this is also the outcome of the visit of His Excellency Tedros Adhanom, the director of WHO.
We have just witnessed the good progress that has already been made, and the WHO is proud to be part of it, and we will continue to give our support. More technology now, not only for Egypt, but we hope you will support Africa and other regions.
So by his visit and his commitment with us, and also by the commitment of Biogeneric and with the Egyptian government, we start to enhance the system at our facility. And to comply with all of that, we put a very comprehensive quality system, because it's one of the mandatory, as the vaccine industry is very, very difficult, and by the end, the quality of the vaccine is very crucial. We are going to vaccinate healthy baby, so we should be sure of the quality of the vaccine to be produced. So we work for product quality, and we comply with ISO 9001 for the employment and the health safety for 45,000 ISO, for environmental 14,000, and for information security 27,000. Why information security? Because everything in the organization is fully automated. We have all the system, brand new, fully automated system. So also we take care of data integrity. And now data integrity, one of the most crucial point while we had the inspection coming from WHO or FDA. This is an overview of the product which is produced in our facility. So through technology transfer, we had about monoclonal antibodies and vaccine product. The registered product, as you see on the screen, so we have rabies vaccine, we have polio vaccine inactivated, we have finglomod, it's therapeutic product, and we have bevacizumab, which is used for some cancer diseases. And also, this is a product which is going to be under registration. So we have filgrastim, enoxaparin, insulin with different full range of the insulin. And in the pipeline for the technology transfer, we have hexavalent vaccine, flu vaccine, yellow fever vaccine, and meningitis vaccine. And this is an ambitious plan. Hopefully will be ended those products which in the pipeline by 2027. So, to complete the journey, this is a manufacturing facility for drug product. So you will see from the screen that we have two lines, fill and finish, and for pre-formulation and formulation. So the first line is the robotic line, 3-in-1, which is going to produce 3 types of presentation. So it will produce pre-filled syringe, cartridge, and vial. And as you see, for the pre-filled syringes, it's the huge capacity. So it is about 7,000 syringes per hour. So the annual capacity about 30 million syringes per hour per year. And for the other line, which is the vial line, you will see we produce in different presentations. So 2R, 6R, and 10R, 50R. So different vial volume. So it can comply with the requirement if I have a vaccine in a single dose or in a multiple dose. And as you all know that multiple dose is the mandatory for the EBI program in each country to facilitate the logistics, and also to be the price can be compromised for the multiple dose. So this is an example of the capacity for the 2R vial, which is a single dose. The capacity is about 76 million vial per year. And for the vial multiple dose, it's about 70 million vial, which is equivalent to 700 million doses of the vaccine can be produced annually. So this is for the phase 1 of our facility, and this phase has been finished, and the GMP had been licensed from our Egyptian Drug Authority. For drug substance production, so the area is about 1,400 meters square, and this area had been constructed, but not yet finished because of lack of funding for this area. So as you see, now if we are going to talk about the platform for messenger RNA, what we have as BGB, we have drug product area constructed, certified and licensed by the drug authority in the country. And I can say BGB spent about more than 50 million US dollar in the construction of this area. It's not only the production area, but also the supporting area, which is the QC, which is the cold store, to maintain the cold chain management. All the people, they are looking for the manufacturing, it is a facility only, but we should take care of the storage area, because cold chain management is the major aspect for the vaccine quality. So we finished phase 1, and we spent almost 50 million US dollar in this area. We construct drug substance area, but not yet finalized due to lack of fund. This is for QC and R&D. As you see, we allocate about 1,100 meters square for this area. And for QC, it's cover the activity of quality control to monoclonal antibody, vaccine, cell and gene therapeutic, and excipient adjuvant. We dedicate an area for R&D, mainly for messenger RNA vaccine. So we construct the lab, we equip it by furniture. And also you will see within BGB, as I mentioned, that we have everything electronic. So this is the roadmap or the mind map for the IT system. So we have Oracle system, everything automated in the system for the filling line, for LEMS, for the lab, for QMS, everything is integrated, and by the end, it's connected by Oracle. We invest in construction of the facility, but for human resource also, we work very hard to give them a training. So this is the training journey through 4 years. As you see, how many training we give it for the people inside and outside. It is related to the GMB concept. It is GMB based on risk assessment and biomanufacturing process for the vaccine. And this is some of the external training which had been conducted last year. So you see upstream vaccine manufacturing, messenger RNA manufacturing and analysis, also biomanufacturing training for workforce empowerment and technology transfer training. So this is most of the critical training which had been given to the staff, because we invest a lot in the staff. It's very important in the vaccine production, not only in the equipment, but also in the staff. And now by the end, what we are doing now for this project, we dedicate the lab, as I mentioned, for messenger R&D and R&D activity. So this lab had been dedicated for an area of 65 meters square. This area had been fully equipped with the furniture and some equipment. So as you see here, this is the activity which had been conducted to messenger RNA project. So you see for the COVID or the platform for messenger RNA, we get the training for lab skills on technology at Afrigen. It was 2022. Technology transfer document had been already also for 1 ml scale. It was in December 2022. And the gap analysis had been identified and sent to MBB. R&D lab established at BGB. It was November 2023. And allocation of the fund, thanks for MPP, they allocate about $500,000 to start the 1 ml activity. And equipment acquisition, we initially initiate the URSs, BRBO, and it was by June 2024. Collaboration, we signed a collaboration agreement with Sheffield University in UK. And this is some of the potential vaccine which we are thinking to work on for messenger RNA. So we have the rabies vaccine, flu vaccine, malaria, Rift Valley, TB, and yellow fever. Some of them urgently needed in the country, and the other for the African continents. I can say for rabies vaccine in 2017, the country consumption was at that time 500,000. And now we use 7 million doses of the vaccine. So we have increased in the number of the doses needed for the vaccines. That's why we think to have it under the technology of messenger RNA, because the method now for the production of rabies vaccine is a long journey to produce it through the routine now which is doing. So if it is, can have the technology to produce through messenger RNA, I think the time will be faster and we can have a lot of doses very fast. This is some of the milestone which was very important for us. So after signing the agreement with MPP, so we had the visit, as you have seen early in the presentation for Tadros Adhanom. It was in October. In January 2023, messenger RNA technology transfer visit coming from WHO and also engaged by AMRO office and MPP and Egyptian Drug Authority. In February 2024, we have assessment by MPP followed by a report, assessment report. It was in April 2024. And in October 2024, we signed the work plan for 1 ml and 100 ml. So this is, in summary, the essential milestone which we are, which was for this project. Oh, sorry. And for R&D lab at BGB, the current status is that the equipment is partially equipped for CNC facility controlled environment for furniture installed. Documentation, critical documentation had been issued, and it's comply with ISO standard, so it is within our scope in the certification. And this is some of the equipment. Oh, sorry. And for the team, this is a structure for the team which is working under this project. So you will see we involve a lot of department working on that because we need to have, it is a cross-functional team working under this project. And coming to the question, when you have the fund, at which time, what is the time frame you are going to produce? If it is, we have a product and we need to have emergency license, we can say it is within 20 to 18 months. But if it is a regular one, it takes more because of the clinical trial, preclinical, it takes more. Finally, this is the last and the most important slide for us. So this is the budget needed for R&D and lab activity. So we calculated it's about $4 million in order to complete the R&D facility for the production messenger RNA. We talk about 1 ml, 100 ml. So we ask for 4 million and unfortunately we receive only $500,000. We ask the support from many institute. We scale up this to the government. But the point is that still up to now, they ask to submit feasibility study. For the feasibility study, we should identify a product. And then when I should identify a product, I can say when the return on investment. So it's a very hard question to answer. So that's why we need the help for MPP with WHO in that, in order to make it easier for us. By the end, this is a challenge and conclusion from our side. So firstly, we succeed to award a small fund coming from MPP. So it is 1 ml for 1 ml, but it's not enough. So this is a challenging facing us. It is a fund. So for R&D activity, we ask for 4 million. We have only $500,000, so the rest will be 3.5. And to establish 100 ml scale, we need 1 million for GMP manufacturing for drug substance. As I mentioned earlier in my presentation, that we construct the facility already. But to finish as GMP, we ask for 9 million. So for the CTD, for the clinical trial, preclinical trial, so we calculated it will be about $2.5 million. So still we have a gap in the fund. We need the help, we need to have the support in this in order to go forward. As I mentioned that we had success pillar in my country. The first one, I can say the drug product facility is already licensed. We have a surplus capacity. We can produce different type of vaccine. As well, we have a very important milestone also in the country that the regulatory authority is maturity level 3, had been announced in February 2023. So it is maturity level 3, and now they prepare for maturity level 4. So it's, I can say that if we are going to produce a vaccine, we have a very strong regulatory authority. So the support will be highly needed for us. So thank you very much. And if you have any question.
Thank you very much indeed, Hiba. So thank you for keeping more or less to time, all of you. So we still got about 10 minutes before lunch. So if you have questions for any of our speakers, and I will prioritize our partners who are online because they don't have the opportunity to come up and talk to you during lunch. So if there are any questions from Polyvac or Inceptor or NIH in Pakistan or Danitza or Torlak, please feel free to ask them. And in the meanwhile, if you just put your hand up if you're in the room, and okay, well, I have a question that if nobody's putting their hands up. So this is I asked a difficult question of Karen, so I think it's only fair that I should ask difficult questions of you. So what I'd like to know from each of you is how you see your government support. So this is not about criticizing your government, but it's more about here's an opportunity to make an ask of your government. If there are areas where you think definitely they could do something to support you, and whether that's changing policies to prioritize local manufacturing in terms of actually buying the products, whether it's in terms of funding, whether it's in terms of providing support from academic institutions within the country. What would you like to see your government do more of? So yeah, I'll start with you, Abraham.
Is this on? Yes. There we go. Thanks so much for that question, Charles. So I think broadly speaking, I think since COVID, there's been a large push for R&D activities to happen within the context of Africa. I think similarly, it's happened in Southeast Asia and in countries around South America as well. So I think from a goodwill point of view, there's a lot of partnering companies that want to work with low middle income setting companies and get projects to a point. I do think, however, from a local manufacturing point of view, what we really need to see a lot more of happening is really the incentivization of these projects, right? Looking at making R&D capability stronger in Africa or in any lower middle income setting for that matter, but really through the incentivization also meeting these partners halfway. So in other words, for example, we do a lot of work with quite a lot of the global funding organizations such as the Gates Foundation, Wellcome Trust, etc. And quite often they're looking at partners also to kind of give an in-kind contribution in terms of projects. And so I think this is where local government for sure can make a major contribution in terms of pushing these partnerships a little bit further and incentivizing it. And then I think it goes without saying, once you have products, you have to sell the products. And so I think the AVMA incentivization is a key stepping stone to moving it that way. But also once local manufacturing has been established, to maintain it and to keep it going takes a lot of money. And so I do think from a local point of view, if, again, support for these initiatives, workforce development can happen, it would go a long way in terms of establishing sustainable vaccine manufacturing on the continent. Those are some of my comments.
Thank you so much, Charles. I think generally speaking as well, but Indonesian government has to fully support our initiative to establish our capability in producing mRNA vaccines from field to finish, from across of the line of production, from drug substance to fill and finish as well. As biopharma, also the state-owned company, there is a lot of funding from government as well that has been invested as well in our facility as well. One thing that I can see in the future is I think government try to also leverage the local university, as currently at the moment we have a huge gap between the university and the manufacturing So Biopharma is quite experienced with the vaccine manufacturing process, but the local university, in the research itself, we are lack of some experience. Like there is some gap how to bridging the translation from the university to manufacturing, and mostly our product from Biopharma is based on the transfer technology from abroad. So we would like to see more the candidate coming from our local university, and perhaps the government will invest more further in that side.
Thanks, Indra. And yes, this is also an opportunity to actually thank your government if they're doing a really fantastic job. So feel free to do that. Now, I can't see if there are people online with questions.
There is actually a hand up online.
Ah.
Maria.
Can I come to you in a minute? Maria, do you want to go ahead?
Yes, thank you. I just have a question, in particularly for Biopharma, but as well for the others manufacturers. In the studies that they have of the market and vaccine demand, what is the main market that they are looking for the production? They are looking for local demand, to cover local demand, or to cover other markets. What are the main objectives for them?
I think we are looking for both internationally and local as well, as most of the product for the local we are willing to we would like to support the government program for vaccinations. Let's say if the next government has a new vaccine introduction like MR or HPV, we would like to support the government by producing the DS locally as well, because we have the huge number of people. So I think it's quite fair to be able to strengthen our capacity in that. But also we are looking for the international product. Since we collaborate with CEPI, we are willing to produce the CEPI's target vaccines disease as well. So we can leverage our mRNA facility in case there is an outbreak in other region. Let's say there is outbreak in African continents or Latin America. As we part of the vaccine manufacturing network from CEPI, we would like also to produce that. So I think we both support local and also international demand for that.
Thank you. And I have a second question. I'm sorry, I want to get the opportunity. If any of them are looking for improve this platform regards the thermostability or regards the efficacy in humans to get vaccines that are more with immunity, with more duration, thermostability and duration of immunity in humans. They are two topics that still remains and needs to be improved in this platform. And if any of these manufacturers are working on these topics, trying to improve and to get better conditions for the countries that are receiving these products. Thank you.
So I think it's obviously it's the big question when we're talking mRNA, right? Thermal stability, I think, is a massive question. Everybody's asking about it, everybody's talking about it, especially within the context, again, of lower middle income settings. Now, I do think that there's a lot of manufacturers and companies working on this. If I'm not mistaken, there's even been some success. I think the latest mRNA vaccine candidate out of Moderna, I think the RSV also stabilizes the product a little bit more and possibly talks about cold chain distribution at 2 to 8 degrees Celsius. So I think from a scientific point of view, I think there's been leaps made. But to answer your question specifically, within the context of where the research stands today, I think there's a lot more to be done in terms of thermal stability, just in terms of being able to move this vaccine to where it's needed quickly. And I do think that there's a lot of programs working on it. BioVac in particular, we're working also on another program together with the Gates Foundation on mRNA specifically. And one of the key aspects that needs to be addressed will be thermal stability on this program. So do I have the perfect answer? Unfortunately not, but it's something that everybody, I think, is putting a lot of emphasis and attention on.
Would you like?
Thank you.
Okay. So from Biopharma side, we see there is a lot of aspect that we can improve in terms of thermal stability and also efficacy as well. But at the moment, we try to establish our capability as the established method first. Then we try to, once we are already confident with our capability, then we will try to explore another technology like new formulations, perhaps new construct of mRNA. There is some university already start working with this gluer RNA. Then perhaps after we capable to establish the established one, then we will seek for the next one. Yeah.
All right, I'm going to bring it to a close now as it's past. So thank you very much indeed to all of our speakers. It's amazing what progress you're making. Well done. And in terms of funding being provided to partners, it's not us. We're just channeling. It's actually coming almost entirely from Canada and from France. So they're the ones who need thanking rather than us.